7 U.S. states have already taken an extraordinary step. They have made it illegal to sell a new type of meat now being developed in laboratories. And yet, at the same time, federal regulators are clearing similar products to appear on grocery store shelves across the country. That contradiction alone should make any thoughtful person pause for a moment and ask a simple question. If this technology is completely safe, why are some governments choosing to ban it while others are approving it? Today, we're going to look carefully at what this new food actually is and what happens at the cellular level when it is produced. I'm Dr. Michael Evans. For years, I've studied how nutrition, metabolism, and aging interact, especially in people over the age of 50. In this video, we're going to examine the science behind cultivated meat, the questions researchers are still asking, and why this topic deserves serious attention before it becomes a routine part of our daily diet. At the center of the entire cultivated meat debate is a question that sounds simple but carries enormous scientific weight. What actually happens inside the human body when we regularly consume cells that were engineered, selected, or adapted to divide continuously? Not for a few weeks, not for a short regulatory trial, but over 10, 20, or even 30 years of normal daily consumption. The honest answer from the current scientific literature is straightforward. We do not yet know. No long-term human population has been followed for decades, consuming cultivated meat as a regular part of their diet. No epidemiological study has tracked cancer rates, metabolic markers, or chronic disease outcomes over a full generation of exposure. The technology itself simply has not existed long enough for that type of data to exist. Most of the safety evaluations conducted so far have focused on short-term biological testing. Researchers examine whether the cells themselves appear stable, whether contamination risks are controlled, and whether the final product meets existing food safety standards. Those are important steps, but they are not the same thing as understanding long-term biological effects in humans, and that difference matters. When a new technology enters the food system, especially one involving living cells grown outside the body, the key scientific question is not only whether the product can be produced safely today. The deeper question is how regular exposure interacts with human biology over time. That is the question scientists are still trying to answer. And before we can even begin discussing potential risks or benefits, we first need to understand something much more basic: what cultivated meat actually is at the cellular level. Before we can evaluate the debate around cultivated meat, we need to understand exactly what it is at the biological level. Despite how it is often described in media headlines, cultivated meat is not a plant-based substitute, and it is not a soy or fungal protein product designed to imitate meat. The material itself comes directly from real animal cells. The process typically begins by taking a small sample of cells from an animal. These are often muscle satellite cells or fat precursor cells. In a living animal, these cells normally help repair tissue or maintain muscle structure. In cultivated meat production, those same cells are removed and placed into a controlled laboratory environment. Inside large stainless steel tanks called bioreactors, the cells are suspended in a nutrient-rich liquid medium. This liquid contains the basic building blocks cells need to grow: amino acids, glucose, vitamins, minerals, and specific growth signals that encourage the cells to multiply. Under the right conditions, the cells divide repeatedly and begin forming strands of tissue that resemble muscle fibers. Over time, these growing cell populations accumulate into material that looks structurally similar to conventional meat. The ultimate goal of companies developing this technology is to scale that process to industrial levels so that large volumes of meat-like tissue can be produced inside facilities rather than on farms. On paper, the idea appears elegant. It promises a new method of producing animal protein without raising entire animals. But once we move beyond the concept and examine the biology more closely, an important question appears. For this system to work at commercial scale, the cells inside those bioreactors must behave very differently from how cells normally behave in nature. To understand why scientists pay so much attention to how cells behave in laboratory environments, we need to start with a basic rule of biology. In healthy organisms, cells are not designed to divide forever. Every normal cell carries a built-in biological limit on how many times it can copy itself. This limit is known as the Hayflick limit, named after the researcher who first described it. Under normal conditions, most cells can only divide a specific number of times before they stop replicating. After reaching that threshold, the cell enters a state called senescence, where it remains alive but no longer continues dividing. This limitation is not a flaw in biology. In fact, it is one of the body's most important safety mechanisms. By restricting how many times cells can replicate, the body reduces the risk that damaged DNA will accumulate across generations of cells. In other words, the Hayflick limit acts as a protective brake on uncontrolled growth. When cells lose that brake and begin dividing indefinitely, the situation becomes biologically significant. In medical science, continuous, unrestricted cell division is one of the defining characteristics of cancer cells. Tumor cells essentially bypass the normal regulatory systems that stop healthy cells from multiplying endlessly. This distinction matters when we look at large-scale cell culture systems. In nature, muscle cells inside an animal follow the normal rules of cell biology, including this limit on replication. But in a bioreactor designed to produce cultivated meat at industrial scale, the economic goal is exactly the opposite. The cells must continue dividing again and again, producing large amounts of tissue over extended production cycles. Once we understand the natural limits placed on healthy cells, another question quickly emerges. If most cells are biologically programmed to stop dividing after a certain number of replications, how can cultivated meat production generate the enormous quantities of tissue required for commercial food markets? The answer lies in something researchers call immortalized cell lines. In large-scale cell culture systems, companies need populations of cells that can continue dividing far beyond the normal Hayflick limit. Instead of stopping after a limited number of replications, these cells are selected or adapted so they can keep multiplying over long periods of time. This allows a relatively small initial sample of cells to produce large amounts of biological material inside a bioreactor. There are several ways scientists can achieve this. In some cases, cells are genetically modified so that the internal signals that normally stop division are altered. In other situations, cells are simply cultured repeatedly over long periods until naturally occurring mutations appear that allow some cells to continue replicating longer than others. Over time, those faster dividing cells become the dominant population. From a manufacturing perspective, this approach makes sense. Continuous cell division allows the system to generate the volume required for industrial food production. But biologically, the process raises important questions. The mechanisms that allow cells to bypass their natural replication limits are closely related to the same regulatory pathways that control growth and cell cycle stability inside the human body. And those pathways are among the most intensely studied areas in modern cancer biology. To understand why scientists pay such close attention to how cells divide, we need to look at the genetic systems that regulate cell growth. Among the many genes involved in this process, two stand out as particularly important: TP53 and PTEN. These genes are often described as tumor suppressors because they act as safeguards that prevent cells from growing or dividing in ways that could become dangerous. Let's begin with TP53. In medical research, TP53 is frequently called the guardian of the genome. That description reflects its role inside nearly every cell in the human body. When DNA becomes damaged, whether from environmental stress, radiation, toxins, or simple errors that occur during normal cell division, TP53 is one of the first molecular systems to respond. It can temporarily stop the cell cycle to allow repair mechanisms to correct the problem. If the damage cannot be repaired, TP53 can trigger apoptosis, a controlled process that causes the cell to destroy itself rather than pass faulty genetic information to future generations of cells. The second gene, PTEN, plays a different but equally critical role. PTEN helps regulate signals that control how quickly cells grow and divide. It acts as a braking mechanism within cellular signaling pathways, particularly those connected to the PI3K pathway that promotes cell survival and proliferation. When PTEN function is reduced or lost, cells may receive constant signals to keep growing. Because of these roles, both TP53 and PTEN are among the most studied genes in cancer biology. They form part of the complex regulatory network that keeps cell division under careful control. Once we understand how important genes like TP53 and PTEN are in controlling cell growth, the next logical step is to ask what happens when cells are grown for long periods in laboratory environments. This question has become increasingly important as cultivated meat technology has expanded from small experimental systems to industrial-scale production models. Several scientific papers have examined the biological challenges of expanding animal cells outside the body. One area researchers often discuss is genetic drift, a process that can occur when cells are repeatedly cultured over time. In simple terms, every round of cell division carries a small chance of mutation. In normal tissues inside the body, many protective systems exist to correct these errors or eliminate damaged cells. But in artificial culture environments, the conditions are very different. When large populations of cells are grown continuously in nutrient-rich environments designed to promote rapid replication, certain cells may gain a growth advantage if mutations allow them to divide faster or survive longer than neighboring cells. Over multiple generations, those cells can gradually dominate the culture. Scientists studying cell culture systems have long recognized this effect. In laboratory research fields such as biotechnology and regenerative medicine, strict quality control protocols are often used to monitor genetic stability in cultured cell lines. These checks help ensure that the cells being used maintain predictable biological behavior. As cultivated meat production scales up, similar questions naturally arise. Researchers are continuing to investigate how cultured animal cells behave over extended periods and how genetic stability can be monitored in large production systems designed to operate continuously. As cultivated meat technology has moved from laboratory research into early commercial production, one issue continues to appear in scientific discussions: the absence of long-term human consumption data. This point has been acknowledged directly in several academic papers examining the future of cellular agriculture. Researchers have noted that while the biological processes used to grow animal cells outside the body are well understood in biomedical science, using those cells as a large-scale food source introduces a different set of questions. Food safety evaluations typically focus on immediate risks, such as contamination, toxicity, or allergic reactions. Those are important considerations, and regulatory agencies routinely evaluate them when new food technologies are introduced. However, long-term dietary exposure is a different matter. For most foods that have been part of human diets for centuries, long historical experience provides a large body of informal safety data. In contrast, cultivated meat is a relatively new technology. Commercial availability began only recently, meaning that no population has consumed it regularly over the span of decades. Some researchers have described this situation as a knowledge gap. It does not necessarily imply that the technology is unsafe. Rather, it reflects the simple reality that the necessary long-term data does not yet exist. Observational studies that track health outcomes across many years require time to develop. Because of this, discussions about cultivated meat often include two parallel perspectives. On one hand, scientists acknowledge the technological promise of producing animal protein through cell culture. On the other hand, they also emphasize that a complete understanding of long-term dietary effects will take years of continued research and monitoring. Up to this point, we have focused on the biology of cell culture and the current state of research around cultivated meat. But there is another dimension that often receives less attention in public discussions, and that is the role of age. The potential implications of any new food technology are not always identical across all age groups. Human biology changes over time, and those changes can influence how the body responds to different environmental factors, including diet. As people move into their 50s, 60s, and beyond, several normal biological processes begin to shift. One of the most important involves the gradual accumulation of genetic changes within cells. Every time a cell divides, there is a small chance that a replication error can occur. Most of these errors are corrected by the body's repair systems, but over decades of life, some changes inevitably accumulate. This process is a normal part of biological aging. At the same time, the systems responsible for detecting and repairing DNA damage can become slightly less efficient. Proteins involved in maintaining genetic stability, including mechanisms associated with tumor suppressor genes, may not function with the same reliability they had earlier in life. This is one reason why the incidence of many cancers rises with age. None of this means that aging itself is a disease. It simply means that the biological landscape of the body becomes more complex over time. Because of that, when evaluating new dietary technologies, many researchers believe it is important to consider how long-term exposure might interact with aging biology in different populations. Another important aspect of cultivated meat production involves the signals that encourage cells to grow and divide inside bioreactors. In living organisms, cell growth is not random. It is carefully regulated by a network of biochemical signals known as growth factors. These molecules act like instructions, telling cells when to divide, when to repair tissue, and when to slow down or stop multiplying. In normal physiology, growth factors are released in response to very specific situations. For example, the body may increase certain signals during wound healing, muscle repair after exercise, or periods of development. Once the repair process is complete, the signaling typically decreases and the tissue returns to a stable state. This balance helps maintain normal cellular behavior. In laboratory cell culture systems, the situation is different. Cells placed in bioreactors must be encouraged to multiply continuously in order to produce sufficient tissue for commercial production. To achieve this, the culture environment often includes growth signals that help maintain active cell division. Among the most commonly discussed molecules in this context are insulin-like growth factor, 1-IGF-1, fibroblast growth factor, and epidermal growth factor. These compounds are widely studied in biology because they influence pathways that regulate cell survival, metabolism, and proliferation. For researchers developing cultivated meat, growth factors help maintain healthy cell cultures and ensure that the cells continue replicating long enough to produce usable tissue. At the same time, scientists continue studying how these signaling pathways function in different biological contexts, including nutrition and aging research. Because growth factors play such a central role in cell biology, scientists have spent decades studying how they influence human health. One of the most widely researched molecules in this area is insulin-like growth factor 1, commonly known as IGF-1. This compound helps regulate growth, tissue repair, and metabolism throughout the body. During childhood and adolescence, IGF-1 is essential for normal development. It supports bone growth, muscle formation, and many other physiological processes. In adults, however, the balance of growth signaling becomes more complex. The body still uses IGF-1 for maintenance and repair, but the levels and timing of that signaling must remain carefully regulated. Over the past several decades, epidemiological studies have examined whether circulating levels of IGF-1 are associated with different health outcomes. Some large population studies have found correlations between elevated IGF-1 levels and increased risk of certain cancers, including prostate, breast, and colorectal cancer. Researchers believe these associations may be linked to how IGF-1 interacts with cellular signaling pathways. When IGF-1 binds to its receptor, it can activate intracellular systems, such as the PI3K, AKT, mTOR pathway, which helps promote cell survival and proliferation. In healthy tissues, this pathway is tightly controlled, helping cells grow or repair themselves when necessary. The important point in the scientific literature is not that IGF-1 is inherently harmful. In fact, it performs many beneficial functions in the body. Rather, the key issue is balance. Because of its role in cell growth and survival, researchers continue to study how long-term patterns of growth factor signaling influence aging, metabolism, and disease risk across different populations. While much of the public discussion around cultivated meat focuses on technology and environmental impact, another important dimension involves nutritional complexity. Traditional animal foods are not simply sources of protein. They contain a wide range of compounds that interact with human biology in ways that researchers are still continuing to explore. For example, meat from animals raised on pasture often contains higher levels of certain fatty acids and micronutrients compared with animals raised in more intensive feeding systems. One compound that has received attention in nutrition research is conjugated linoleic acid, commonly abbreviated as CLA. Some studies suggest that CLA may influence inflammatory pathways and metabolic regulation, although research in this area is still evolving. Another nutrient often discussed in connection with animal foods is vitamin K2, particularly the form known as MK4. Vitamin K2 plays a role in activating proteins involved in calcium metabolism, helping guide calcium into bone tissue while preventing unwanted deposition in soft tissues such as blood vessel walls. This function has drawn increasing interest in research on bone health and cardiovascular aging. Animal foods also contain structural proteins such as collagen and amino acids like glycine and proline, which contribute to connective tissue maintenance in the body. These nutrients are naturally present in many traditional foods that humans have consumed for generations. The broader point is that whole foods often provide nutrients in complex combinations that work together biologically. As new food technologies develop, researchers continue examining how different production methods may influence the final nutritional profile of the foods people eventually consume. Another topic that has entered the discussion around cultivated meat involves the environment in which these cells are grown. Unlike traditional livestock agriculture, where muscle tissue develops inside a living animal, cultivated meat is produced inside industrial bioreactors, large, highly controlled containers designed to support continuous cell growth. These systems are typically constructed using stainless steel tanks, plastic tubing, filtration systems, and in some cases synthetic scaffolding materials that help cells organize into tissue-like structures. The presence of plastic components in modern industrial systems is not unique to cultivated meat. Plastics are widely used throughout food processing, medical manufacturing, and biotechnology because they are lightweight, flexible, and resistant to contamination. However, in recent years, scientists have begun studying a related issue that has drawn increasing attention: microplastics. Microplastics are extremely small fragments of plastic that can form as materials break down over time. Environmental research has detected microplastic particles in many parts of the natural world, including oceans, soil, and even atmospheric dust. More recently, several studies have also identified microplastics in human tissues, such as blood samples and lung tissue, although researchers are still working to understand the full implications of these findings. Because cultivated meat is produced in controlled industrial environments that may involve plastic surfaces, tubing, or scaffolds, some scientists have raised questions about whether microscopic plastic particles could interact with cell cultures during production. At this stage, research into this topic is still developing. Scientists continue to study how materials used in bioreactor systems interact with biological cells and how production environments influence the final purity of cultured tissues. As with many aspects of new technologies, the presence of open scientific questions does not necessarily indicate a problem. Rather, it highlights the importance of ongoing research, careful monitoring, and transparent investigation as the field continues to evolve. As cultivated meat technology continues to develop, it is also important to understand the broader economic environment surrounding it. Like many emerging technologies, cellular agriculture has attracted significant investment from a wide range of organizations. These include venture capital firms, technology entrepreneurs, and even some of the largest traditional meat companies in the world. Several major food corporations have invested in cultivated meat startups as part of their long-term strategy. For established companies in the global protein market, this investment approach can serve as a way to explore potential future shifts in food production while maintaining their existing business models. By supporting new technologies early, these companies position themselves to adapt if consumer demand or regulatory frameworks change over time. In addition to traditional food companies, a number of technology investors have also shown interest in the field. The promise of producing animal protein without raising entire animals has generated excitement among investors who see the potential for large-scale transformation in agriculture and food manufacturing. Billions of dollars have been directed into research facilities, pilot production plants, and startup companies working to refine the technology. However, financial momentum can sometimes move faster than scientific consensus. This pattern has appeared repeatedly throughout the history of technological innovation. Investors often fund promising ideas years before the long-term outcomes are fully understood. In many cases, that investment accelerates valuable progress. At the same time, it can also create pressure to bring products to market quickly in order to justify the large amounts of capital involved. Understanding this financial landscape helps explain why cultivated meat has advanced so rapidly from laboratory research to early commercial approval in several countries. Another important issue in the discussion around cultivated meat involves transparency. In many areas of science and technology, new inventions are protected through patents. When a company files a patent, it must publicly disclose detailed information about how the technology works. Those disclosures become part of the scientific record, allowing researchers, regulators, and even competitors to study the methods involved. However, not all companies choose that path. In some cases, businesses protect their processes through trade secrets instead. A trade secret allows a company to keep the technical details of its production methods private. As long as the information is not publicly revealed, it can remain protected indefinitely. In the field of cultivated meat, several companies have reportedly chosen this approach for certain parts of their production systems. From a business perspective, this decision is understandable. Developing stable cell lines, optimizing growth media, and designing efficient bioreactor systems can require years of research and large financial investments. Companies may want to protect that intellectual property from competitors. At the same time, the use of trade secrets raises questions about how much information is available for independent scientific review. When production methods remain confidential, outside researchers have limited ability to examine the exact processes used to grow and maintain the cells that eventually become food products. Regulatory agencies typically review safety documentation provided by the companies themselves, but the level of public disclosure may vary. As the cultivated meat industry continues to grow, discussions about transparency, intellectual property, and scientific access are likely to remain an important part of the broader conversation surrounding this emerging technology. When new technologies enter the food system, history often provides useful perspective. Many products that are now common in modern diets were once introduced with great enthusiasm and strong confidence in their safety. Over time, however, the scientific understanding of their long-term health effects sometimes changed as more data became available. One widely discussed example is trans fats. When partially hydrogenated oils were introduced in the early 20th century, they were promoted as a practical and stable alternative to traditional animal fats. Food manufacturers adopted them quickly because they extended shelf life and improved the texture of processed foods. For decades, these oils were used in baked goods, snacks, and restaurant cooking across much of the world. It was only many years later, after large epidemiological studies accumulated, that researchers began linking high trans fat consumption with increased risk of cardiovascular disease. Eventually, regulatory agencies moved to restrict or eliminate them from the food supply. Another example, often cited in nutrition discussions, is high fructose corn syrup. Introduced as a cost-effective sweetener in the 1970s, it rapidly became common in beverages and packaged foods. While the compound itself is chemically similar to other sugars, long-term research on metabolic health later raised questions about patterns of excessive sugar consumption and their relationship to obesity, fatty liver disease, and insulin resistance. These examples do not imply that every new food technology will produce negative outcomes. Many innovations have improved food safety and nutrition dramatically. The larger lesson is simply that understanding the full impact of dietary changes often requires years of observation. As new products appear, continued research and careful monitoring remain essential to building a clear scientific picture After exploring the scientific questions surrounding cultivated meat, it is also useful to look at something much more practical: the types of foods that have long been associated with supporting healthy cellular function, particularly in people over the age of 50. In many cases, these foods are not new or exotic. They are traditional ingredients that have been part of human diets for generations and are increasingly being studied in modern nutritional research. One example is grass-fed beef. Meat from cattle raised primarily on pasture often contains higher levels of certain fatty acids, including conjugated linoleic acid, or CLA. Some studies suggest that CLA may influence inflammatory signaling and metabolic balance, though research continues to explore its full biological role. Another widely discussed food in this context is bone broth, which is produced by simmering bones and connective tissue for extended periods. This process releases amino acids such as glycine and proline that are involved in connective tissue maintenance and collagen formation within the body. Beef liver is often described as one of the most nutrient-dense foods available. It provides high concentrations of vitamin B12, iron, copper, and other micronutrients that play roles in red blood cell production and energy metabolism. For individuals who tolerate organ meats, even small servings can provide substantial nutritional value. In addition, small fatty fish like sardines, mackerel, and anchovies are rich sources of omega-3 fatty acids, particularly EPA and DHA, These compounds are widely studied for their effects on inflammation and cardiovascular health. When consumed as part of a balanced diet, these traditional foods can contribute to a diverse nutritional foundation that supports overall metabolic stability as the body ages. Understanding which foods may support long-term health is only part of the conversation. The more important question for most people is how to apply that information in daily life without feeling overwhelmed. One of the most common mistakes people make when trying to improve their diet is attempting to change everything at once. Sudden, dramatic shifts often lead to short bursts of motivation, followed by frustration when the routine becomes difficult to maintain. In clinical practice and nutrition research, a more sustainable approach is usually recommended. Rather than transforming an entire diet overnight, many health professionals suggest identifying 2 or 3 specific habits that can realistically be maintained over time. For example, a person might begin by adding a serving of fatty fish to their weekly meals, incorporating fermented vegetables into lunches, or replacing highly processed snacks with whole foods that provide more stable energy. Consistency matters far more than intensity. The body's metabolic systems, including the gut microbiome, mitochondrial energy production, and inflammatory regulation, respond gradually to dietary patterns. These biological changes occur over weeks and months rather than days. A steady routine allows the body time to adapt and helps reinforce habits that can last for years. For individuals over 50, this steady approach may be especially valuable. As metabolism changes with age, maintaining stable blood sugar levels, balanced nutrient intake, and adequate protein consumption becomes increasingly important. Small adjustments repeated consistently can produce meaningful improvements in overall well-being. By focusing on gradual, realistic changes instead of extreme short-term plans, people are more likely to build dietary patterns that support long-term health and resilience as they age. When people begin improving their diet, one of the most common questions is how quickly they should expect to notice results. Modern culture often promotes the idea of rapid transformation, but the reality of human biology is very different. Most meaningful physiological changes occur gradually as the body adapts to new nutritional patterns. In the first 2 to 3 weeks, many individuals report improvements in daily energy stability. Instead of experiencing sharp fluctuations in blood sugar that lead to afternoon fatigue, the body begins to rely more consistently on balanced nutrient intake. Some people also notice improved digestion as the gut microbiome adjusts to increased consumption of whole foods, fiber, and fermented ingredients. After 4 to 6 weeks, additional changes may begin to appear. Skin hydration, hair strength, and recovery after physical activity often improve as the body receives a steadier supply of essential nutrients, amino acids, and healthy fats. These visible changes reflect deeper biological processes occurring within tissues that constantly renew themselves, such as skin and the lining of the digestive system. More substantial improvements typically occur over longer periods. At around 3 to 4 months, some people experience measurable changes in metabolic markers when tested through routine blood panels. Inflammatory markers may decrease, cholesterol profiles can improve, and overall energy metabolism often becomes more stable. These timelines illustrate an important principle. The body does not respond to nutrition overnight. Instead, health improvements emerge through consistent patterns repeated day after day. By maintaining balanced dietary habits over time, individuals give their cells and metabolic systems the conditions needed to function. After examining the biology, the research, and the broader context surrounding cultivated meat, it is important to step back and summarize what the current scientific landscape actually tells us. In discussions about emerging technologies, the most responsible position is often one that recognizes both the potential benefits and the unanswered questions. At this moment, cultivated meat has not been conclusively demonstrated to be dangerous. Early safety evaluations conducted by regulatory agencies focus on contamination control, manufacturing standards, and the stability of the final product. Those evaluations are designed to ensure that the food entering the market meets existing safety frameworks. However, it is equally true that cultivated meat has not yet been studied across the long timescales that normally shape our understanding of how a food interacts with human health. Long-term dietary studies require many years of observation. Because this technology is still relatively new, that body of evidence is still developing. In addition, The biological processes involved in growing animal cells outside the body, such as extended cell culture and the use of growth signaling environments, touch on areas of biology that scientists continue to study in fields ranging from regenerative medicine to oncology. These connections do not necessarily indicate risk, but they do explain why researchers remain interested in understanding how these systems behave over time. For now, the most accurate summary is one of ongoing investigation. Cultivated meat represents a promising technological development, yet, like many innovations, its full long-term implications will only become clear through continued research, transparent data, and careful monitoring as the technology matures.